Hi, my name is Nick Banovich, and I'm from the Translational Genomics Research Institute in Phoenix, and I'm excited to share with you guys my work looking for gene expression signatures associated with a novel drug treatment against a cellular inhibitor of apoptosis. So in our experiment, we're looking at a novel agent, LCL-161. It's gone through some clinical trials in solid tumors, but it's also been shown to have anti-tumor activity in multiple myeloma. Now, what's interesting about this treatment is our in vitro work from our collaborators at Mayo Clinic have demonstrated that the treatment, rather than having direct tumor killing ability, actually acts to activate macrophages, and the macrophages phagocytose the tumor. However, it's unclear what the molecular mechanisms underlying this anti-tumor activity are. So we wanted to use a novel technology, single-cell RNA sequencing, facilitated by the 10x Genomics platform, to investigate the underlying gene expression signatures associated with this treatment response. So ultimately, we looked at 18 different mice, and these mice were either had a transplantable tumor and were treated with the two standard of care treatment drugs, either cyclophosphamide or bortezomib. They were treated with the novel agent, or they remained untreated. We also had two control mice, or two classes of control mice, mice that did not have the tumor transplanted and mice that did not have the tumor transplanted but were also treated with the drug. We ultimately performed single-cell RNA sequencing on the spleen from these mice, and the spleen was harvested 24 hours after drug treatment. Ultimately, we ended up with 98,000 cells, and I show these cells here in a 2D representation in what's called a TS&E plot. And this TS&E plot reduces the variation down into 2 dimensions, so we can look at these in a sort of graphical representation. So here, each dot is a cell, and what we've done is use mathematical methods to cluster these cells together by transcriptional similarity using a graph-based approach that's implemented in the R package Seurat. So here, each color represents a group of cells with a similar transcriptional profile. And what we're able to do is overlay marker gene expression, seen here in the example of T cells, and we're able to pull out marker gene expression and annotate each one of these unsupervised clusters into a distinct cell type. So now I'm showing you here all of the distinct cell types that are now classified by our gene expression signatures. So we were interested in the myeloid population, as this population houses the macrophages, which we believe are driving the anti-tumor activity. So what we've done is we pulled out the myeloid population from this larger subset of cells, and we've reanalyzed them only looking at this population. And you can see here the myeloid population colored by treatment condition, and then you can also see the myeloid pseudo-colored by the different clustering using our graph-based clustering approach, and I've also shown you the marker gene expression for one of the macrophage markers, LYZ2. Now, what we were really interested in is we know from some of our in vitro work that you need both the tumor and the macrophages to see the anti-tumor activity. If you just treat the macrophages with the drug and put them in with the tumor, they do not phagocytose the tumor. And so we believe the model is the drug goes into the tumor secretes a factor, the macrophages take up that factor and start phagocytosing the tumors. And so we really wanted to look at gene expression signatures within just these two cell populations of the myeloid cells from the mice that had the tumor and were treated with the drug, and the mice that did not have the tumor and were treated with the drug, and see if we could identify gene expression changes that were specific to macrophages in mice with the tumor and were also treated with the drug. And so what I'm showing here is the same myeloid population, where I've now just pulled out these two different categories. So what you can see here in yellow is, again, I've pulled out the myeloid population, and what I've done is I've pseudo-colored these so that the yellow cells indicate lack of expression, and the red cells indicate an increase of expression. And what we were able to identify was a number of genes that were exclusively expressed in macrophages from mice with the tumor that were also treated with the drug. And now I've also pulled this out into the broader population of cells, both in this box plot and in this TS&E plot. And what you can see here is that the gene expression is really limited only to these cells and these conditions across the entire experiment. And so the reason that we're interested in this is that we're hoping that we can use these genes as potential new targets for therapeutics. As I mentioned earlier, the pathway now that we believe is happening is that this drug that has relatively broad-based expression changes across a number of cell types comes into the tumors, makes the tumors excrete a factor. That factor is taken up by the macrophages, and the macrophages begin to phagocytose the tumor. Ideally, we could target just the genes within the macrophages that are involved with the increase in phagocytosis, and that treatment would be much more targeted and have much higher efficacy. And so we hope to follow up these experiments with additional work to try and see which of these genes is actually the causative genes for tumor phagocytosis, and if we can develop novel treatments that target these genes rather than the broader transcriptional profile. So I want to thank you guys for taking the time to listen to my poster, and if you want more information, please check out my website either by going to tgen.org or banovichlab.org, and I hope you guys enjoyed the presentation.